Medical students often face an overwhelming cognitive load when attempting to memorize thousands of distinct infectious disease symptoms as isolated facts. Many cases present as localized, pyogenic infections, such as focal staphylococcal skin abscesses or endocarditis. Other presentations are systemic and vector-borne, like the widespread symptoms of Lyme borreliosis transmitted by deer ticks. Some pathogens produce highly distinct mucosal signs, such as the primary canker that characterizes early syphilis. Attempting to process these diverse clinical signs as unorganized lists leads to diagnostic errors when overlapping symptoms appear. This master taxonomy tree categorizes every bug by its physical traits, offering a systematic alternative to rote memorization. It functions as a diagnostic algorithm based on cellular structure. Knowing these properties predicts its clinical behavior within the host. Tracing these structural traits informs the selection of antibiotics capable of penetrating the pathogen's specific defenses. Diagnosis relies on the execution of this rigid structural algorithm. The taxonomic tree begins with the bacterial envelope, the primary division of cellular architecture. This diagram of a prokaryotic cell highlights the capsule, cell wall, and cytoplasmic membrane, which together form the cell's main defense against the environment. Gram-positive bacteria possess a thick single layer of peptidoglycan that retains crystal violet stain during laboratory testing. Gram-negative architecture consists of a thin peptidoglycan layer situated between two separate phospholipid membranes. This structural dichotomy dictates the classes of medications required to breach the cell's outer defenses. Identifying the envelope structure immediately narrows the list of potential pathogens by half. The next taxonomic node determines the physical shape of the organism, classifying them as either cocci or bacilli. Cocci are spherical structures that often form grape-like clusters or long chains. Bacilli are elongated, rod-shaped structures adapted for different survival environments. A foundational heuristic rule links these shapes to their likely gram stain results. In the majority of clinical cases, spherical cocci test as gram-positive. Most rod-shaped bacilli test as gram-negative. Combining morphology with a gram stain provides a probable pathogen profile before lab cultures are finalized. Clinicians must watch for specific organisms that violate these general morphological rules. The Neisseria family is the primary exception to the first rule, representing the only clinically significant gram-negative cocci. Identifying red-stained gram-negative diplococci inside host cells is the diagnostic proof for a Neisseria infection. The exceptions to the second rule, the gram-positive bacilli, are tracked using the mnemonic ABCCLLCP, representing organisms like Clostridium, Listeria, and Bacillus anthracis. Recognizing an organism that violates the standard shape-to-stain correlation helps isolate the specific culprit. These structural exceptions turn a standard lab report into a precise diagnostic tool. Certain organisms lack standard structural components, like a cell wall, entirely. Obligate intracellular bacteria are defined by their inability to produce their own ATP. Because they cannot generate energy, these organisms must invade and hijack human host cells to survive and replicate. Antibiotics that target cell wall synthesis are ineffective against these pathogens, since the target stricture does not exist. Mycobacteria, such as those causing tuberculosis, utilize complex waxy lipid walls that require acid-fast staining rather than a standard gram stain. The host immune system responds to these resistant pathogens by forming necrotizing, caseating granulomas to physically isolate the infection within the tissue. When a pathogen lacks typical structural vulnerabilities, the resulting disease is often defined by the host's attempts to wall it off. Host defense mechanisms determine how effectively a pathogen's structural advantages are neutralized. Mapping clinical immune deficiencies against the taxonomy tree reveals the specific types of opportunistic infections that follow. A lack of T cells, seen in DeGeorge syndrome, leads to a predictable susceptibility to fungal and viral threats. NADPH oxidase deficiency in neutrophils prevents the destruction of certain microbes, leading to chronic granulomatous disease. Recurrent infections by the Neisseria family are often the direct result of a missing C6-C8 complement protein. Specific immune mechanism failures correlate with vulnerabilities to exact classes of pathogens. Clinical symptoms reflect specific interactions between the host and the invader. These symptoms reflect the predictable outcomes of a pathogen's physical structure meeting a specific host immune defense. The taxonomic flowchart provides the logical framework needed to navigate complex clinical presentations. Mastering this structural algorithm allows a clinician to arrive at a diagnosis based on the pathogen's biological traits rather than memorized lists.